From Microbe TV, this is Immune, episode number 38, recorded on November 24, 2020. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, welcome back.
How things in Ithaca? Do you have snow yet?
We did. We had a covering. It melted, so no snow currently. But it's actually like 50 degrees today. It's very, very unusually warm for this time of year. I don't complain. I've been going for walks, so it's been quite nice. Also
joining us from Durham, North Carolina, Steph Langle.
Hey there. Good to be back. Another episode. To think that eventually we'll start talking about other viruses than COVID-19, but that seems to be the hot topic.
You know, that's a good question. Like when? You know, you could talk about this for years, actually, because, you know, even after vaccines, people are going to be studying vaccine responses and all kinds of stuff. And from Madison, New Jersey, Brianne Barker.
Hi, great to be here. Having a great time here. It sounds like it might be slightly warmer in Ithaca than in New Jersey, which is odd. But
that is odd.
Yeah, but that's okay.
I'm not going to complain. You can complain.
No, it's okay.
You'll get your laughs back when
I spent enough time in New York to know how exciting that is.
Our guest today is also from Durham, North Carolina. She is the Davison Distinguished Professor of Pediatrics at Duke Medical Center. Sallie Permar, welcome to Immune.
Thank you, Vincent, and thank you to this esteemed audience here and hosts of Immune. I'm, like I told the host earlier, I am just so honored to be one of your guests, and I've really been a fan of your work. Many of your hosts I know very well, and I'm just excited to be here and speaking with you.
Great. You know, well, it's funny to hear that, and it's, you know, it gives me a little like pinpricks. It's very sweet because I, as a PhD student, started reading Sallie's papers, and there's a little teaser before talking about other viruses. We are going to talk about other viruses today, but I was reading Sallie's papers and thinking, wow, wouldn't it be great someday if I could end up in Sallie Permar's lab? And in fact, that's what happened. Sallie is my boss, and I've been in the lab since 2019.
Yeah, it's not very long.
No, it's not. With the pandemic, I mean, there's like...
It just feels like 2020's been...
It feels like forever, yeah.
Yeah.
It does feel like forever. And I still remember when I had joined, I was probably new on Twitter, pushed there by my husband, who's a PR guy. And so I was glad to be joining. And I was quickly followed by a Steph Langle, who I thought, wow, she's really excited about breast milk immunity, mucosal immunity, maternal immunity. And that's how I got to know her. And then I visited Ohio State University, gave a talk there, and Steph came over from the vet school like an hour away, right?
Yeah, I was at a branch campus, which is where we did a majority of our work. We have BSL-2 and 3 ag facilities, so they require like tons of space. And we're up there, and I did drive down because I had to make sure to come down. Now, who hosted you that weekend?
It was Jessie Quick.
Jessie Quick, right.
Who had previously been at UNC, and we had interacted through HIV research at UNC.
Perfect.
But after meeting Steph, I thought, well, if she applies, definitely taking her as a person.
That's very nice. And this doesn't have to be like a Steph and Sallie fest. So although I'm enjoying this very much.
We could let you guys talk. It would be okay.
We'll just have a side conversation.
You know, when we heard all the stages of Steph's career, because when she joined Immune, she started it with us. She was a PhD student, and we heard her think about postdocs, and then Sallie, your name came up, and then she joined your lab. And so we're getting a history of Steph on Immune.
Real time.
Real time.
She's a great role model.
Oh,
so when you interviewed her, you knew she was doing this podcast, right, Sallie?
It came up somewhere along the way. I remember Steph said, now I have to let you know about my other life that I pursue on social media and in science communication, and she wanted to make sure I was okay with it because it is a time commitment.
Yeah.
But I am a believer that there is a lot of benefit to anyone's career who pursues science communications on some level for their own work, to promote their own work, promote those of others, that I'm okay with it. So I'm glad that Steph is really an example, I think, in our group and how to utilize social media for good. And so I watched her doing that and I learned from her, really.
You've been very involved in a lot of different capacities, which I think we can talk about in a little bit. But I've seen write-ups, the New York Times or the LA Times. I can't remember which one, but a lot of different publications.
I have been, I say, inspired by this pandemic on some level because it really was a culmination of all the things that us as virologists, immunologists, we've been talking about and writing about in those few critical intro sentences of our grants. Like, the reason we do this work is that, viruses are a problem, and we know there are more coming. We know that there are, the high potential for these things to become epidemic, even pandemic. But when it happened, it felt like even we weren't prepared, and it felt like it brought together so much of my world, not just the science, but also how much it affected us as professionals, as mothers, parents, how, to explain to your friends what was going on, vaccinologists. And so I was inspired to write and talk to the world, as many people have been. And so I put my hand in on editorials and op-eds and opinion pieces that I hadn't done before. I'm really inspired by the pandemic.
So, Sallie, let's start with a little bit of your history. Where are you from originally?
I am from right here in Raleigh, North Carolina, the Raleigh-Durham area. So I was a public school kid from Raleigh, North Carolina. Neither of my parents were scientists, but I had some, very life-changing moments with teachers in my science courses throughout high school that made me think maybe science was for me, and went to Davidson College, which is a small liberal arts school outside Charlotte, where I solidified my interest in research. From there, one thing that I did in college, maybe that I can recommend to others once we're not under travel advisories, is I took the opportunity to go outside of North Carolina, which I really had not left for most of my life at that point. And I just felt like I needed to get out. And so I signed up for a summer experience in Mwandi, Zambia, a very far remote end of the world, where only recently had they gotten electricity out to the hospital that was there. Wow. And it was the mid-90s when the AIDS epidemic on that continent was really at its peak, and there was no treatment. And that really had an impact on me and what I decided to do with my career.
So when you graduated, did you immediately have your next step planned?
Somewhat. And then there were lots of pivots along the way. But yes, I came home from that trip because as I was there and thinking about all the suffering and really a missing generation of parents was really what was apparent there. There were lots of children who were sick from the virus. And then there were grandparents who were taking care of them. And it was, and their parents were sort of, wasting away, many of them. And so I thought, well, how can we address this situation? It seems so dire. And I thought, well, a vaccine is what would address this situation. And so I went home from that trip thinking, I want to work on vaccines, and decided to go apply for PhD programs in immunology as my next step. And
you did that at Johns Hopkins, right?
Yes. And while at Johns Hopkins, I had a wonderful mentor, or actually it was a coincidence how I found my mentor, that I was so intrigued by my experience in Zambia during just a short summer experience that I thought, well, I'd like to go spend more time there and really pursue research in earnest that takes more time than a summer. And so I signed up for several fellowships, or applied to several fellowships, ended up getting a Fulbright fellowship that I was able to go back to the University of Zambia and spend an additional year. And I was working in the virology lab. And I had a great study planned. The Kaposi sarcoma virus had recently been described as a cause of, an AIDS-related cancer. And there was this pediatrician in the University of Zambia, Professor Chintu, who was the department chair of pediatrics, actually, in the University of Zambia. And he had written a paper about how this virus may be transmitted from mother to child because he was seeing pediatric cases. And so I thought, I remember the conclusions of one of his papers that said more research needs to be done to understand if this virus can be transmitted from mother to child. And I turned that into a proposal. And I remember mailing him a letter to say, would you be my mentor? And he mailed me a letter back that said, yes. So
one word,
clutched
yes.
that letter. Yes. It was maybe a few more words than that, but I clutched that letter. And, when I received the Fulbright based on that proposal, I arrived in country basically holding a letter out saying, like, I need to know where Professor Chintu is. And found him, and he said, oh, great, we have lots of patients.
Wow.
Did you bring all your reagents? And I'll get you the blood samples. And I thought, like, I just got out of college.
Oh my gosh.
I think I know how to do PCR, maybe, but I didn't bring any primers or.
Oh, that's crazy.
And so we realized that there was not going to be a lot I could do on the project I had proposed, but there were other projects that I could, learn from more experienced virologists. And I ended up working on a study that was led by Hopkins, studying measles in HIV-infected children and how it presented differently. And that's where I found my mentor, that my mentor was actually working at the same virology lab in the University Teaching Hospital of in Lusaka. And so I started my PhD project really in that year, and then went on to the PhD in immunology and microbiology at Hopkins.
And that was with Diane Griffin? Is that who?
That is Diane Griffin.
Okay.
Yes.
Okay.
A world-renowned measles expert.
Right.
And she is a physician scientist as well.
Oh, okay.
She was a neurologist by training and a virologist in her research, and really led, much of the work in describing who was still at risk for measles even after the vaccine was available, what we could do to get around maternal antibody interference that interferes with, measles vaccine being completely effective at the young ages. And it was really with Diane that I had my next pivot where I saw physician scientists working with her. They were pediatric physician scientists who were infectious disease fellows in training, and they were also working in the laboratory. And so they would go see a patient with RSV, a baby in the hospital with RSV, and then they would come back and study RSV in the laboratory. And I thought, like, wow, that is really cool. I want to be able to do that. And so I went to Diane during my first year of graduate school and said, I think I want to go to med school. And luckily, I think her being, having that experience herself of having both degrees, she said, that's a great idea. So instead of saying, oh, you can't go to med school in the middle of a PhD, she said, that's a great idea. I hadn't even taken the MCAT. So I said, I'm going to have to take some time off to study for the MCAT. So somehow made it work, applied to medical school in the middle of, or that first year of my PhD, and ended up finishing 2 years of my PhD and then going to medical school. However, not at Hopkins because I didn't get in, but I ended up going to Harvard. And when I went to Diane and thought, okay, if I'm going to go to medical school in a different city, first I said, maybe I should just get my master's because that was an option because I could still do research with a master's. And she said, nope, having the MD PhD was so important to my career. That's what you should do. And so she really supported me. And I said, well, can we find me a new lab in Boston? And she said, okay, that's a good idea. Let's find you a new lab to work in Boston so you can keep the work going while you're in med school. And that's where I was introduced to Brianne's mentor.
Nice. Look at this coming full circle.
In fact, I was excited to hear the first how you got first introduced to measles because when I met Sallie, Sallie was the person who had sort of brought this measles project to the lab. And Sallie was sort of like the measles project in the lab when I joined later on.
Oh, nice.
I think everyone was a little nervous. It was their first respiratory virus, maybe, that had been studied in the lab. And yes, so the tie-in with Norm Letvin, who was a mentor to both Brianne and I, was that he worked on non-human primate models. models and of AIDS. He had discovered the SIV virus and that it caused an AIDS-like illness in monkeys. And so, had lots of work going on in vaccines and pathogenesis there.
And that was a great story of how he discovered it, but perhaps not for air.
Yes, it was a competition between a few investigators, including Norm.
Yeah.
So that is, one to discuss over a beer sometime, probably.
So you and Brianne actually physically overlapped in that lab.
We did.
Wow.
Yeah.
Cool.
So, I think because you were there both as a finishing grad school and then when you were sort of a postdoc and when you were finishing med school.
Yes.
So I started a rotation in the lab in the summer of 2003.
And I think, so I preceded you maybe a little bit. So I was there maybe 2001 or so.
Yeah.
Yes.
And I was there till 2009.
And I was there till 2011.
So it's basically a lot of time we were both there together.
Were your benches close to each other? Were they far away?
They were. I remember when Norm asked me, where do you want to sit? He had several rooms and we walked through and in one room he said, this is the fun room where all the grad students hang out. And I said, well, I want the fun room, obviously. And so I got put in with the rest of the graduate students. Brianne joined eventually.
Yes, I did.
Cool. Very cool. So can I ask you a measles question? This is in the middle of your story, but I just don't want to forget this. Is it true that the measles vaccine is sterilizing? Induces sterilizing immunity?
Sterilizing immunity. I think the answer is no.
Okay.
I think that it doesn't allow systemic virus replication.
Do you know if it allows shedding from the respiratory tract?
That's a good question from the respiratory tract. I'm not sure that has been established.
Yeah.
But one thing we did do in monkeys that we might have snuck into a paper is took monkeys who had HIV and had some immune suppression and gave them a measles vaccine. And we did find some shedding, if I can remember the paper right. So it was a bit controversial to say, like, would it be safe still, to give this vaccine? I think clearly the epidemiology has shown yes, that it has been safe. But in the monkeys, we wondered a little about, how much virus replication could you get, especially, in this immune compromised model.
Yeah. I mean, I ask because we're talking about SARS-CoV-2 vaccines and whether there'll be sterilizing, probably not. And people are always saying around me, the measles vaccine is a great example of a sterilizing vaccine. And I said, really? I'm not sure about that. So that's why
maybe we don't have the evidence.
No. Okay.
Yeah. I wouldn't put it in that category.
Yeah, because vaccines I know the best, the polio vaccines are absolutely not sterilizing, right? So
no,
it's just when they tested the polio vaccines, it was to prevent polio, not to prevent shedding. So makes perfect sense.
And some of the best immunity, which I think is not the same with SARS-CoV-2, some of the best immunity is from natural infection, right? And so, sometimes I think a vaccine that can allow a little bit of natural virus exposure combined with your vaccine immunity that then blocks it from, becoming a problem, maybe some of the best vaccines. But in the SARS-CoV-2 model, we're seeing that natural immunity is actually probably not better than what can be elicited by a vaccine.
Yeah. Yeah. Okay. Sorry. So we're at you finished an MD and then you did a little bit more in Vin's lab. Is that right?
Yes. So he gratefully allowed me to do some measles studies in his non-human primates, and then even in some HIV-infected non-human primates. And we actually did some classic studies that were very Norm Letvin-esque, was to deplete parts of the immune system and then give the monkey the virus and see, what happened. And so he had led some of those studies for HIV, demonstrating that CD8 T cells are very important for virus control. And I sometimes laugh that we keep learning this over and over and over again, that CD8 T cells are very important for control of HIV, maybe not for protection against acquisition, but definitely for control. And we did that in measles, and we showed that the CD8 T cell depleted animals did have a harder time controlling virus replication when they were naive. Yet when you depleted B cells, you had did not have any change in the viral dynamics. So really, the measles infection clearance is dependent on a CD8 T cell immune response. Right. But I stayed on, and I remember having a existential crisis. And this was actually one of the most remarkable things I think Norm did for me as a mentee was he said, okay, Sallie, you're finishing all of your schooling. You're finishing your graduate school, your MD, your PhD. You need to pick an area that's going to be yours, and it's got to be different than mine. And this is an important moment for you. And so he said, I'm going to help you figure out what you want to do, because I had been studying, respiratory viruses, measles. I was in an HIV lab. What did I want to do? And he went with me to maybe 3 or 4, very astute virologists and immunologists around Harvard Med School campus. And we sat in those people's office for an hour or so and just shot the breeze about, if you were Sallie, Norm would ask them, what would you do for your career? What virus or problem would you focus on? Wow. And I think back to that he spent that much time and effort helping me find my career. It's really, really impressive. And unfortunately, I thought that was the standard in mentoring, and I learned later it wasn't. But I really appreciated that. And after doing all that, going around to all these people, thinking about all the opportunities, I came back to Norm and I said, you know what? I want to study pediatric HIV and how we can prevent mother-to-child transmission of HIV. And he said, great, because I can help you with that. We study HIV and non-human primate models, and maybe we can work on non-human primate models for maternal transmission. So we worked on a breast milk transmission model and, studying immunity in humans in the mother-to-child setting as well.
So before you left, did you do some experiments to get some data on that system?
Yes. So we, one of my things that maybe won Steph Langel's interest was I helped to develop a model in which you could induce lactation without the monkey ever being pregnant.
I remember the lab meetings.
Were people like, what are you going to do?
They're like, milking monkeys, huh?
Yes. And there was a whole time in my life, in fact, I'm just donating these supplies to Steph now, where I worked on developing a breast pump for monkeys. Because we weren't getting a lot of milk from the monkeys, and I wanted more. We want more milk all the time to do the cellular immune work and the antibody work and the virus work. And so we thought, okay, maybe the manual approach that the vets that was not, the best it could be. So we tried to build a monkey breast pump. And what we learned from it is that the manual approach by very experienced veterinary staff was actually the best. I did some monkey milking myself, and I, proved that to be true as well. So we stuck with the manual approach in the end. My monkey breast milk pump never came to be, and, never resulted in a paper either.
Could have
but
been
yes,
a big seller,
everyone had to put up with that. Hearing about that.
So after Harvard, where did you go next?
So then I was out of my fellowship. So I trained in pediatrics and then pediatric infectious disease. One important thing that I think I was able to do for my career, because it is a lot of time that you commit to clinical training, and that's a lot of time you can be out of a research group or out of a lab that you may lose ground. And we see that a lot of people who have an interest, say in medical school and research, and then they go into clinical training and just don't find the time or the mentorship, and then they may never go back to it. In fact, only 1% of doctors describe themselves as researchers, which I think should be higher. And so one thing that I did was I did an integrated research residency where I combined laboratory research with my clinical training in pediatrics, and then pediatric infectious disease fellowship as well. And it was nice that I stayed at the same location and with the same mentor, and that I had such a wonderful mentor and such wonderful lab mates, of course, Brienne, that I stayed there and kept working.
Yeah, that story that you were telling about Norm going to different meetings with you is such a Norm story.
Aww.
And, I can think about sort of similar experiences that I had, and,
well, that's good, because I was thinking it's like
very reminiscent as you were telling that.
I was thinking that. I was like, oh, I hope Brienne also had that experience. Otherwise, this could be awkward.
Oh, no, I very much did.
Oh, that's good. That's good.
Very much. And Sallie and I have, had conversations since about how much, it would be great to have him around to bounce ideas off of now.
Yeah.
Yes. Because as Brianna indicated, he passed away in 2012, right? Yeah. I believe, from pancreatic cancer, which he actually was first diagnosed with in 2008.
Right? Yeah.
So lived a long time with that that disease, which usually it's much quicker than that to people succumb to it. But he what really kept him going throughout that time was he kept working. He kept meeting with mentees. I mean, I met with him maybe 2 months before he died, and I remember him telling me, I was telling him about my CMV work, which we can get to, and I told him about trying to develop a model of congenital CMV, and he said, "Sallie, that's a good idea." And like, I still remember that. And it was like 2 months before he passed away. So there's still like moments from Norm that pop up in my head, and like things little sayings he had that still guide me today.
Yeah, no, my students have all learned many normisms that I still repeat. And just like Sallie, I think I last met with him about 2 months before he died. And I was telling him about deciding to be at primarily in undergrad institutions teaching, and he's like, "Good, you're happy." And that was sort of the last thing that he said. So
good, you're happy. I'm glad you said
someone
you're
who
happy.
had a big influence on
yes,
of us.
Huge influence.
Nice. And so that you were going to tell us where you went next, I think, right?
Yes, I'm almost to Duke now. Yeah.
On the road to Duke.
On the road to Duke. So yes, I went through my training. I was on faculty. Norm fought hard to, say, "Sallie, I had received a K award soon after my clinical training." And he said, "Sallie, you are at the level of an assistant professor." He put me up for assistant professor. Whole thing getting set up there as a junior faculty. And he gave me one of his very great technicians that I started working with. He said he handpicked someone for me. It was so wonderful. And then I had a tragedy happen in my life where my sister passed away from a early colon cancer. One of those early colon cancers that maybe sometime we can discuss. What is that? People in their 40s getting really bad colon cancer. I feel like, and it's a rising incidence compared to, the older age groups for colon cancer. I feel like it's a virus, but maybe someone smarter than me can figure that out. But anyway, I said, I need to move back to North Carolina. My sister left 3 young girls, and I wanted to go back, and my family was on board also because I had a newborn at that time. And so we want to go back to North Carolina and help raise my nieces. And so Norm said, "Okay, I will call my buddy at Duke University, Bart Haynes, who leads the Vaccine Institute there." And Bart and Norm were colleagues and had worked in the first version of the Center for HIV AIDS Vaccine Immunology, CHAVI. One of the largest grant that the NIH had ever given was to Bart Haynes and a group of investigators, Norm being a key one.
Yeah.
And I had become part of that work. Bran probably was even part of that work.
I was.
We were all on board. And so it was nice that Duke was able to recruit me from there. So that was my first faculty job. And I had a nice transition again because my mentor really set me up with the right next set of mentors.
Yeah, I mean, the two of them have worked together so well throughout so much of their career. I was actually an undergrad in Bart's lab, and he called Norm and said, "I have this grad student."
Oh, look at that.
Great for you. So
that's so funny. I think a theme is that, having good mentors is key. And so when I talk to, grad students or people at other universities and they're trying to find their way, it's like, okay, if you're not one person can give you maybe everything you need, although maybe it seems like Norm was doing that for everyone. But you need to diversify your mentor, the people you look towards, because their networks are only, they're so big, and then your network only grows by their network. So, I think that's a key thing that we're learning about here.
Yeah.
So if not for your family situation, you might have stayed at Harvard most of your career, right?
I might have. My husband was a Bostonian, and I was happy at Harvard, and things were going well. When I got to Duke, I did. It was like a breath of fresh air having one medical school, one campus, a lot of very friendly people, as Southerners generally are. And, I think it was a great place for me to start a career, very collaborative. I think, with a system like Harvard it's very siloed kind of, and there's a lot of, I think there's challenge for junior faculty to make it through all the potential barriers. And, of course, there's many strategies by which to bolster those early careers. But Duke was a great place for me, and I really got the best of taking advantage of early career programs as well as the collaborations and the work from the Human Vaccine Institute.
So given that we don't have a huge amount of time, I would enjoy if you would, because I want to get to SARS-CoV-2 before we end. Maybe you could give us an overview of the HIV and CMV work you did when you went to Duke, and we can ask you questions about it as you go.
Great. Yeah, so my focus has really been on determining the immune responses that can be protective against neonatal infections that mostly occur from mother-to-child transmission. And there's some lessons from natural immunity to gain there that then can guide vaccine development, which is that for both of those viruses, even when mom becomes infected for the first time during pregnancy, it's only about 30 or 40% chance that the mom is going to pass on the virus to. And so that's a large number who don't get that infection, even though mom has no immunity, the baby has no immunity. And so trying to understand what is it that protects that 70% of exposed moms and neonates against that vertical transmission has been sort of at the heart of what we've tried to study. And so we've done some work establishing some immune correlates of protection against neonatal HIV. It's a virus that, as we've learned again and again, you have to both study the immune system and the virus together because the virus changes so rapidly in response to immune pressures that you can't study one without the other. And so some of our recent work has shown that, no one was ever able to really come up with a specific type of antibody response that may be protective in the mother-child setting when mom is passing the antibodies to the baby before the baby's even born. And we're learning now that it may be because the viruses that infect those babies are actually just escaping those antibody responses. And so if you define a potentially protective antibody response in another setting, it may actually be driving transmission in the mother-child transmission setting. And we're finding that may be true of broadly neutralizing antibodies in that mother-child setting. And for CMV, it's even more complex. I talk about that SARS-CoV-2, even HIV, you only have to focus on one protein when you come to antibody responses. With cytomegalovirus, it's 14 different complexes on the surface of that virus. And so still learning, what are the important responses there. And you also have not only learning about what can prevent maternal acquisition, but also what can prevent the actual placental transfer as well, maybe different immune correlates. And that's where having an animal model has been very useful, and a highly relevant animal model that has a placenta that mirrors that of humans is an opportunity. Still using the deplete one cell type in the immune system to see, what's most important. I feel like HIV, we're still a bit far away from those broadly neutralizing antibodies that I believe we're going to need to have in order to have a protective vaccine, but getting somewhat closer with the native-like antigens. And then in CMV, I think we're closer. We've had protective vaccines that have reached 50% efficacy, and so studying what was successful there and enhancing that may be a way to improve on that.
So, Sallie, I know that you have done a little bit of work on vaccination during pregnancy and vaccination of breastfeeding moms and how the vaccine-elicited response can be transferred to the neonate. Can you talk a little bit about that work? Because I think that's, when I've heard you talk about it, it's been really fascinating.
Yeah, right. So that, again, is one of the things that really draws me to this field of maternal infant immunology is there is the opportunity to use the mother's very mature immune system to elicit responses that then will aid the baby in getting through a very vulnerable time right after birth. That we know that immune system couldn't be too overactive as a fetus because you have to avoid rejecting your own mother as a fetus. And so the immune system has to be modulated in several ways to not do that. And so therefore, you can use the opportunity of the maternal immune system, which includes antibodies passing across the placenta. And of late, we've been studying, does every single IgG get across the placenta at the same rate and with the same chance? Or are there ideas about which antibodies may selectively cross the placenta, and that would allow us to control it a bit more through vaccination? But the other piece of the maternal immune system that's very important is the breastfeeding period. And there is immunity that is transferred to the infant through breastfeeding as well. And so there's really those two opportunities to use to enhance immunity for the baby who is still developing their own immune system.
It's a similar approach to respiratory syncytial virus, right? Immunize the mother because the babies are so susceptible at birth that you can't really vaccinate them, right?
Right, because any immune system, no matter how old, needs a few weeks, right, for the adaptive response to develop. And so even immunizing the minute you're born, it's going to take a few weeks. And those few weeks are when we see the most babies admitted to the hospital with RSV. So yes, that's been a big focus of the RSV vaccine in eliciting protective antibodies that will be specifically. Well, though my OBGYN colleagues tell me, don't forget about the moms. Moms also should not get RSV in pregnancy because that can be a severe disease for them. But really, for the purpose of protecting the infant in that first few weeks of life, maybe combined with active vaccination in the infant, that would generate protective immunity across the ages.
Now, you and your MD PhD student, Jennifer Jenks, just had a recent publication, Science Translational Medicine, correct?
Yes.
Defining a correlate of protection for CMV. And maybe in the next, couple minutes before we transition to SARS-2, could talk about that and what it means for actually getting a licensed CMV vaccine.
Yeah. So as an HIV vaccine researcher, I came to the field of CMV really because in my pediatric infectious disease training, I was working in clinic and I saw baby after baby who was coming in for evaluation of congenital CMV. And I thought, who's working on this vaccine? That's literally what went through my head. And so then I went and looked it up, and it seemed like it was an open area that didn't have near as much activity going on as the HIV vaccine, and yet was so common. 1 out of every 150 babies born across the globe has congenital CMV, and about 20% of those will go on to have some kind of long-lasting impairment. Hearing loss being the most common, but other developmental delays, other birth defects as well. It's the rubella of our time, or of all times, because it's been with us, as long as we know. So it is important to develop a cytomegalovirus vaccine, and I think it's one that's really going to help things like the education gap, because it is a disease of poverty and in certain underrepresented minorities where it's more common in those populations. So very important vaccine, and it's actually one that's caught the eye of the vaccine manufacturers because it's needed across the globe, developed countries and underdeveloped countries. So it was actually Moderna's lead candidate before SARS-CoV-2. And so it's still in there.
SARS-CoV-2 is stealing the spotlight.
It is, but I think it's blazing a path as well.
Okay. Yeah.
So we set out. So I was learning about the CMV field. It was remarkable to me that they had one vaccine candidate, the glycoprotein B vaccine, which is the main fusion protein of CMV, mixed with a squalene adjuvant. That had been tried in 3 different populations in postpartum women who are going to be exposed to their toddlers who often are carrying CMV, adolescent women who are going to be the target population of the CMV vaccine to have immunity before pregnancy, and then in transplant patients, because CMV is also the leading transplant infectious complication. And in all 3 of those studies, it was remarkable to me, they achieved approximately 50% efficacy in preventing acquisition, or in the kidney transplant, preventing reactivation of the virus. And so I thought, something must be real there if you've tried it in 3 different populations and got the same answer. but no one had looked at the immune correlates. And so a group in the UK was doing that with a kidney transplant group. And we worked with the groups that had done the postpartum and the adolescent vaccine and gathered together all the samples that were available, which was a Herculean task, actually. And then had enough power to do an immune correlate analysis, and really found that something surprising was, while the vaccine was a soluble protein and therefore likely in the post-fusion state, we know the SARS-CoV-2 vaccine is in a pre-fusion state when it's been designed. So this is a post-fusion vaccine that was given. We found that the antibody response, not to the vaccine itself, but the antibody response to the glycoprotein B expressed on the surface of a cell, was actually the immune correlate of protection. And so responding to the native conformation, probably of the pre-fusion CMV-GB, is what was associated with protection.
So since people are infected relatively early in life with CMV, what would be the target age for vaccination? Would it be very young? It would not just be women, I suppose, which would be everyone in theory.
Yeah, that is the $1 million and probably more question for the vaccine manufacturers. So, there's the CMV vaccine, there's CMV transmission to the fetus when mom is infected for the first time. But importantly, and this took a while to get into the field, it's not like rubella, where natural immunity is not protective against the placental transmission. Even those that have been infected before pregnancy can either reactivate the virus or acquire a new strain, because it's not protective immunity. There is reinfection, and that may get transferred. That may get passed to the fetus. So while the vaccine right now is being developed in the seronegative women, which are mostly in developed countries, eventually that vaccine, to really address the worldwide issue of congenital CMV, will have to elicit improved immunity to just the natural immunity that the virus itself elicits. So right now, adolescents are the target, but there's been a lot of talk about, should this be a toddler vaccine, which is also where a lot of the transmission is happening. Back to your question about, can it prevent transmission in addition to acquisition?
Yeah. Great. Wow. I feel like we could talk about these topics for sure, like another hour.
For sure.
I'd be game.
But so you've been able to like pivot from one virus to another. And so I guess you've, like many people, now pivoted to SARS-CoV-2. Is that something that you would like to talk about a little bit and tell us what you're doing in that arena?
Yeah, sure. So yes, I'm not sure I would recommend pivoting from virus to virus, because every time you do, you have to develop the new neutralizing antibody assay and all new reagents. Steph knows, because she's been doing that for rotavirus in our group. But I think it's, as immunologist, these principles apply across all viruses. And so really that's an immunologist's job, right, is to figure out how to study those important pieces of the immune system against many potential pathogens. So it's challenging, and we're always struggling with virus stocks, but I have found that to be enjoyable to attack the next problem. And that really was SARS-CoV-2. I think as any virologist or immunologist, you just felt the weight of, well, what's going to be my place in fighting this pandemic, realizing how impactful it was going to be to, all of our lifetimes. And I'm a little embarrassed that one of my friends in the non-human primate world came to me in January, I think, and he said, "Hey, we're thinking about getting a group together to study this Wuhan virus. Are you in?" And I said, "I'm really busy. I don't know if I can take on another thing," which is good advice usually to people who are already studying a lot of viruses. But at that time, I clearly didn't recognize how important this was going to be. But once it became February and it was pretty apparent how much of an issue this was going to be for the whole world, I started getting interested, and I know others too, on why are children seemingly protected against severe disease and the adults and elderly are filling up the hospitals. Usually there is what we call a bathtub shaped curve for respiratory viruses in terms of severity, heavy on the young and on the elderly. And this one is clearly just a curve to severity in the elderly. And I still think that understanding why children evade severe disease is key to protecting elderly and really impacting this whole pandemic at its core.
And that's still holding up, like over time. We still, even now schools are reopening and a lot more children are getting sick. They're just not getting severely ill.
That's true. We braced for the wave for a while in pediatric infectious disease. It didn't come. What did come is the multi-inflammatory syndrome in children that really has only presented in earnest in numbers and numbers of children in places that were hard hit all at once, like New York City. And then those of us who were in parts of the country that didn't have that initial very high volume surge, we were all waiting and checking every patient's D-dimer, et cetera, to look for MISC. And we realized it must be pretty rare. So the severe disease is still rare, I would say, in children. I think we think MISC of something like 1 in 1,000 maybe infections. And there are some groups that may have more severe disease, like adolescents, especially obese adolescents. But yeah, that has really held up, that young children seem to maybe have a few mild symptoms, but not need hospitalization.
So do we have any ideas? I know there are tons of things that have been proposed in the literature, but I'd love to hear kind of what data stands out to you for why there might be that difference in severe disease in children. What types of things are you thinking about with that?
Yeah. So I think our hypotheses have not changed, and there's just maybe a little data here and there for each of them that have kind of refined them. But the first being that the virus can't replicate as well. Like maybe all the requirements for entry, et cetera, are just not expressed as highly on the nasal epithelia and the lung epithelia of children. The immune system is stronger or better or more appropriate or not too robust to get that cytokine storm. And then third, that there's some pre-existing immunity that children have that are protecting them, with cross-protective coronavirus responses that adults are less likely to have. And I would say there's some evidence probably for each of those. One thing that has been clear, though, is that children do not replicate the virus less well. We can see really high levels of virus in the nose of children, similar to that of adults, and it doesn't seem to go along with symptoms. But maybe the children are clearing it faster. That's one paper that we published, Matt Kelly and Jillian Hurst led out of Duke, by studying children who did not come to the hospital, did not go to the clinics, but they went to their homes. So adults who were diagnosed, they then called them up and said, "Do you have children?" and went to their homes. So that's a really remarkable cohort that we're still working to describe what happened in completely asymptomatic or very mildly symptomatic children. So maybe there we showed that the virus was contained potentially more quickly in those kids. Then some work out of Einstein and Betsy Harold's group is that maybe the innate immune system is more robust in children and does the job at maybe clearing the virus quicker. And then some recent work that maybe there is that potential for pan-coronavirus antibodies in the B-cell repertoire of children that's more frequent than in adults. And I think that'll be something that's really important for vaccines in the future. Where are we going to target these pan-coronavirus vaccines? Right. And I want to make the argument for pediatric immunity.
So what do you, there's been a big controversy about whether young children transmit at all. They seem to transmit less effectively, but I'm not sure we have enough data. What do you think about that question?
Yeah, that's really been at the crux of it. And I wanted so much to believe that children were not going to transmit as well as adults or at all, ideally, so we could send them back to school because, children are really suffering from not having the safety net, really, that school provides, in addition to the critical, in-person learning. But it didn't pan out that way. Children can transmit. Clearly, household transmission is common. Even in this study that Jillian Hurst and Matt Kelly ran at Duke, a sibling being infected was a risk factor for a child becoming infected. And, there have been lots of clusters where children have been the transmitter. But I think what we've learned from the school experience, and I've been busy trying to gather data on this, although, working with communities and school partners is a challenge into itself, is that if you practice the 3 W's of wear your mask, wash your hands, and wait in line 6 feet apart, then you can prevent the secondary transmission in a cohort setting. So you can't necessarily prevent an infected child coming to a school campus, but it seems that if you are very diligently adhering to those infection control measures, you can prevent the secondary transmissions. And so I think that's the lesson that we've gotten from the school setting.
Yeah. Oh, and those things are such a low, there's not an economic bar for those 3 things, right? And it's sad to me that there are school closings happening because of community transmission where the solution can be done in schools. You can do those 3 W's. There's a disconnect there.
I would say, yes, everyone can wear a mask and wash their hands. I think the challenge, and especially in New York City, per se, is the spacing. So the desks really have to be 6 feet apart? The number of times I have thought about that question is too many. Can they be 3 feet apart? I mean, it makes a big difference. That's twice as many kids in a room if your desk can be 3 feet apart versus 6 feet apart.
You're right. Yeah.
So I think the space has been the biggest issue.
So that you're right. I take back that is an economic issue, right? Because we think about funding of schools and the ability of them to have, the schools that don't have the same type of funding, they have to pack kids in. So that makes sense.
Yeah, I know. Here in Ithaca, they have them in cohorts. So they have 2 cohorts that go in person. And so one cohort goes Monday, Tuesday, another cohort goes Thursday, Friday. Everybody's asynchronous on Wednesday. And then there are the virtual learners. And it's about, if I have the numbers correctly, it's about 1/3 are completely home, at least in our high school. 1/3 are in the first cohort and 1/3 are in the second cohort. So, and that has to be at that level in order to have the distancing here. With a very large high school with a large outdoor campus, like 12 buildings connected together, a courtyard where they can go out and eat, even in winter. I don't know. We'll see how that happens. But yeah, I think that it's difficult for us here, even in this more remote area with fewer students, to handle that kind of thing. I mean, the high school is not particularly small. It's like 12 to 1,600 students. So that's, a good sized high school, but it's a physically large high school. But even then it's difficult. So I can't imagine what being able to do that kind of thing in a city where there's significantly more number of students attending. Right.
Yeah.
I think we still have a problem. Yep.
We do. However, I have been dismayed at that schools have been closed when the places where we know transmission is happening, restaurants and bars, are still open. That is not listening to public health. That is not listening to the scientists. And it obviously
kids.
It's not thinking about kids. It's not acting in the best interest of kids. And of course it has economic impacts. And that's where, our government, if it was working, would be giving stimulus to those, those businesses that are being affected and keep the kids in school. And we have really done a disservice to children through this pandemic. And we're going to be paying for it for years to come.
Yeah.
Yeah. New York City just closed its schools. But it's because they had a contract with the teachers union that above 3%, they would close them. They were trying to do it. And they think transmission is very low in New York City schools, although I'm not sure they're testing enough. Once a month, a fraction of the kids is not enough to know that. But the problem is when the kids get out of school, they forget everything that you've just mentioned, the 3 W's. And, I think that's where transmission occurs.
That's right.
Yeah.
Big problem.
Yeah. So I think if we could go back and do it right, we would get low cost, low sensitivity, even testing to schools. Yeah. We would prioritize schools being open, close other businesses, give them the economic relief they need. And we would have been paying teachers more from the beginning, because I think this was a breaking point for teachers.
Yeah, absolutely.
So giving them hazard pay and then never taking it away would have been different for children.
Well, there's going to be another pandemic in 10, 20 years. You think we'll remember the lessons?
I hope. Let's elect leaders who can remember the lessons.
Oh my gosh.
So.
Okay, so Sallie, and so we're going to, maybe in the last 5 minutes, you can talk about your new exciting position. And I was just going to say, maybe you'll be there for 10 years and next pandemic will hit, you can run for president and then we'll vote for you and then you can put those things into place.
Well, can I ask one more SARS-CoV-2 question first?
Yes.
Okay, so my other question was, what are your thoughts about the vaccine trials and whether or not kids are going to be vaccinated, and how
Thank
the
you, Brianne.
vaccination will work with kids?
That is my platform. Thank you. Because if you hadn't asked it, I would have said, wait, I have one more thing. Yes. So I think another way that our system does not serve kids as well as it could is kids are often left to the last group that we think of in research studies. Same with pregnant women. A lot of it, I think, just trying to do the right thing in terms of safety. They can't consent for themselves.
Right.
At the same time, the paternalistic view is keeping the advancements in science away from children, and it's really kind of unethical in that sense as well. So clearly, kids are clearly being severely impacted by this pandemic, even if they're not in the hospitals because they're not in school. And they are, we're losing generations of time on the education gap, et cetera. So we have a responsibility to get these vaccines to children so that there is no concern about outbreaks from school. And also there are children who do end up in the hospital. We see them every week at Duke, and I'm sure in New York City, they'll be even more frequent, that are severely impacted by the virus. And of course, the multi-inflammatory syndrome that did prove to be very severe. So we have a responsibility to get these innovations to children. They were not the first group, obviously, that was vaccinated in trials, and I agree with that we need to get it to the those that are being most severely impacted, the elderly. Keep the healthcare workers working. But at this point, we know that they are safe and efficacious in adults. They should absolutely be trialed in children, and Pfizer is doing that. They're at least going down to age 12. Moderna is working towards opening younger ages. But the old way of doing it that I think we need to rethink, and now that we do warp speed vaccination, we should rethink this, is the age de-escalation. I don't think there's any scientific basis for that. That first we'll do down to 12, then we'll do down to age 6, then we'll do down to age 3. I don't think there's really a scientific reason for that other than somebody just came up with it. So I think that needs to be rethought. And luckily, we did get funded early on thanks to some champions at the NIH, to study some of the leading vaccine platforms in infant monkeys. Because while we knew the human trials would not happen right away, that was something we could do. And stay tuned, because I do think that infants will respond well to many of the leading candidate vaccines. And we can at least first demonstrate that in a highly relevant model. And then I'm looking forward to those vaccines coming to fruition in the pediatric trials. And we won't make it for school this year, but I hope for everything that we have a vaccine to give them during the 2021-2022 school year.
Yeah. So basically, kids are not getting vaccinated with this first round of vaccines because we haven't trialed them in the kids. And we're going to have to wait until we trial them in the kids, just for our listeners who are not up on all of this.
Thank you. Yes. And I advocate for more quickly including children in vaccine trials.
Did you want to comment on your next, what did you call it? Throughout your career, you've done these
Pivots.
Your next pivot?
There we go. Yes, it's another pivot. Yes, so I have recently taken a role as department chair of pediatrics for Weill Cornell Medical School, as well as the physician-in-chief for New York Presbyterian Weill Cornell Pediatrics. And so this is a pivot for me because it includes overseeing all 3 missions of a academic medical center for pediatrics, including research, clinical care, and teaching, training the next generation of pediatricians. And while I have done a lot of the research end and a lot on the training end, running clinical operations will be the add-on for me. I've been a physician, practicing pediatric infectious disease physician for a while, but overseeing all of the primary care, the specialty care for a lot of the sector of New York City is an exciting opportunity. I'm thrilled to be joining that institution. Really an amazing place to pursue research, and just such an opportunity to make a difference for so many children throughout the 5 boroughs and beyond. And so I'm really thrilled, but it certainly will be a pivot.
Well, you got to be
so
consistent,
lean into those
right?
pivots. Would that be a piece of advice you would say, Sallie?
Surely, because I can tell you that when I arrived at Duke 9 years ago, no way did I think I would ever be a department chair. No way. I was fighting for my life, really, as a junior investigator, trying to get that first grant. And once I got a hang of that part, I did look up and say, well, how can I start to change the system for the next group of people who come through here? Because again, I think there are too few of us, right, who are pursuing this work. And obviously the pandemic caught us off guard. That should be a sign. So I do think I was able to look up from, just writing grant after grant and experiment after experiment to say, how can I potentially change the system? And that's how I got interested in administration. I've led some work in physician scientists at Duke and children's research. And those things really led me to say, but what if I could bring in the clinical operations to say, how can you really pair the advancements in the clinical side with the advancements from the research side, and then put training in the middle of that? And it's really, an amazing opportunity to make a difference in how we administer care, how we, achieve better health and prevention of disease as well.
I can imagine Norm would be super psyched.
Brian, that when I told Bart, our other shared mentor, that's exactly what he said.
Yeah,
He
sure.
said,
Yep.
Well,
Norm is smiling down, and I do
yes.
believe that.
if you ever find yourself up at my neck of the woods, since you're working with New York Presbyterian Hospital, then let me know, and maybe we can record an episode right in my office. That'd be
I would love that.
That'd be like in a year or two when we're starting to do normal things again. Well, good luck with that.
Yeah.
Thank you.
It sounds like
It's a great place to be up there in that area right there with Sloan Kettering,
Upper East
Rockefeller,
Side.
Cornell. Yeah.
Yes.
Hospital.
I was going to say, Cindy and Sallie. So, I mean, I know that they're two separate institutions, but I mean, you would be considered under the same umbrella of Cornell.
Yes.
Yes.
So
Right.
two things. One, that's where I did my PhD. So I know quite well.
Nice.
And two, there is a huge force to try and integrate the two institutions. So there's a lot of money put towards this academic integration between Weill Cornell and Ithaca campuses. So there's a lot of opportunity, especially you should come up and visit the immunology community. We have the Center for Immunology. We don't have to, broadcast all of the wonderful, cool things about Cornell, but there really is a vibrant immunology community connected between the two. That's really exciting. So it'll be welcome to have you.
That's great. I think we've just found our next grant writing endeavor is a shared campus immunology grant, I think.
Nice.
And yes, the NYP also wants to combine its campuses, make them even closer and more in line, especially for pediatrics. So I will be doing a lot with Columbia also.
Great. Looking forward. Well, Sallie Permar, Duke University, thanks so much for joining us. It's been great.
Yeah,
Thank you for having me.
Pleasure.
Wonderful to hear your story.
Well, thank you. Best hour I've spent of many days.
Good. Glad to hear it.
Glad to hear.
Bye-bye.
Thank you all so much.
Bye.
Good to see you, Sallie.
Bye-bye.
Thanks.
Good to see you, Brianne.
Well, that was fun.
That was great.
Yeah. Very interesting career, right?
Yeah, I think there's a lot to be learned about the pivoting, the mentorship.
Oh yeah.
Sallie goes a mile a minute, and so for me as a trainee, I've learned a lot. I mean, it's been sadly a short amount of time that I've been, with her, but I've learned a lot for sure.
I'm sure, but I don't think she's going to bring you to 3 different PIs for an hour each and talk about what you should do next. Right?
You know what's so funny? She said, I was like, I don't know if we've ever talked about that before.
Oh no, that's a very, very Norm thing.
Okay, I'm like,
I can come up with a whole bunch of similar things that happen to me.
Oh, that's good.
Also it's a thing of the past, unfortunately, because the field was smaller and people knew each other more.
Right.
Nowadays, you don't know many people. You can't just walk into someone's office for an hour, and you can't do it. People are too busy, and there are a lot of more of scientists, and we don't know each other. It's too bad in a way, because that's a great way of mentoring, for sure.
Yeah, I mean, the thing that we really had as a benefit was that at that time, they had recently gotten that grant that she mentioned, the Chavi grant, that was so huge. And so that basically meant that Bart and Norm and all of those people in that group didn't have to write grants for a while.
Yeah,
Yeah,
nice.
that's crazy.
And so a lot of the time that they had previously spent writing grants, they devoted towards mentoring us.
Yeah.
That's interesting.
So we were really lucky.
Brianne, when you were in the lab with Sallie, did you know she would do really well one day?
Oh yeah, Sallie was very clearly sort of an all-star in the lab. And I she didn't mention it, but I know that she had some collaborations with some groups in Africa while she was at Harvard. And so one of the things that, I remember her lab meetings talking about milking monkeys, and everyone sort of making different faces. But I also remember once that she was trying to go do some experiments and brought in a suitcase and was like throwing serological pipettes in a suitcase to try to figure out how to have that experience where they said, okay, where are your reagents?
Yeah.
So crazy.
And so, there are lots of little things, but she was always, really fantastic and really clearly going to go very far.
By the way, the experiment she was talking about with non-human primates and measles, where you deplete, B cells and T cells, those must be going on now with SARS-CoV-2, because you can infect.
I would think so.
It would really.
I'm sure. I don't know which group, but
I'm sure they are. And one thing that she sort of didn't mention, whenever I think about that set of experiments that she did with the depletion of B cells and the depletion of CD8 T cells, that is a big experiment I think about when thinking about the difference between what actually is an immune response that's protective in natural immunity versus what can be protective in a vaccine setting. Because those papers showed dramatically the importance of CD8 T cells in controlling infection in natural infection. Yet if you look at the antibody response, that seems to be such an important response in the measles vaccine.
interesting.
And so whenever I think about the fact that a protective immune response in a vaccine, the response that has to be there when the virus comes to the door, is different than the type of response that has to happen to get rid of the virus. The first paper I always think of is that paper of Sallie's.
Interesting. Yeah.
Yeah,
I think,
I always break that down when I teach that. There's immune defenses that prevent new cells from being infected, or like if you're exposed newly, prevents that infection, versus eliminating or already established infection within a cell.
Yeah, you've said that.
They're really
Cindy.
two different things.
Yep, exactly.
And that's why we have two parts of the immune system to complement each other.
That in grad school was kind of where I got it, was from that study.
You have
It's
said that,
beautiful.
Cindy, that if you've already got a bunch of infected cells, the antibody's not going to help too much. So you need CTLs to get rid of them. So it's very interesting. She mentioned Diane Griffin. So when I was a PhD student, the dogma was antibodies prevent infections, which is pretty much, for the most part, still true. And T cells help you recover. But Diane Griffin did an experiment in mice with Synbis virus, which showed that antibodies are actually essential for recovery. It's a mouse model for a disease that doesn't happen in people. But so there are some cases.
I guess the contributing factor would be that antibodies can go intracellular.
In her case, the disease was a nervous system disease. And so she showed that antibodies actually get into the CNS, and and they cure infections somehow. That was Beth Levine's work, by the way.
And also there's this antibody dependent cell cytotoxicity that people kind of ignore, but that can be a major part of defense. So it's not that T cells do this and antibodies do this,
right?
But they can both to some degree help with both parts of the prevention and elimination.
So ADCC is where the antibody and a cell can collaborate to kill an infected cell, right?
So the way I look at it is it works best with a envelope virus, right? Because envelope virus need to decorate the surface with their glycoproteins so that they can bud out.
Right.
But you've got antibodies, the glycoproteins, because that's the way they work, is they bind to the virus. But if the surface of the cell looks like the virus, you bind the antibodies, and then all the other cellular mechanisms that will detect that, macrophages eating that, complement activation, or NK cell activation, they all, the NK cells, they all can attack that infected cell that's now coated with antibody.
Right.
Yes. We're always
It's
remembering
not a very
that.
commonly referred to mechanism, but
Yeah.
It's one of those things that, we need to remember the antibodies do more than just neutralize.
When I teach my slide on how antibodies work, that's part of it for sure. It's not just,
absolutely.
and it's worse that most people think antibodies block uptake of virus into cells. Okay. They think, it makes sense that it blocks, the antibody binds the receptor binding protein and it blocks attachment. It's not the only way, but so many misconceptions.
Yeah.
But that's why we're here to reverse them. Okay. That was a cool episode. Thank you, Steph, for arranging that.
That was
really
really fun.
appreciate it.
Yeah, good. I'm glad you guys had a good time. It was fun.
She's just great. Great conversationalist, really interested and passionate about science. I love it. It's really good.
Good.
All right. That's Immune episode 38. You can find the show notes at microbe.tv/immune. And I remind everyone that there's a new Instagram account for microbe.tv. It's being curated by Lauren. She's doing a great job summarizing every episode of all the podcasts and getting people involved and engaged. And if you have questions or comments, immune@microbe.tv. If you'd like to support us right now through the end of November, if you go over to parasiteswithoutborders.com and donate to them, they're a 501(c), so your donation is tax deductible. They'll match your donation and give it back to us. So that's pretty cool. That's parasiteswithoutborders.com. Cindy Leifer's at Cornell University on Twitter. Cindy Leifer. Thanks, Cindy.
Thank you.
Steph Langle's at Duke University. Stephanie Langle on Twitter. Thanks, Steph.
Yeah, thanks. This was fun.
Brianne Barker's at Drew University. BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
I'm Vincent Racaniello. You can find me at virology.blog. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.